All right. Well, I think it's time to start. I want to start by saying hello to everybody. Greetings, fellow color enthusiasts. Welcome to Color Literacy Forum number 6. I'm Luann Stovall. I'm the host of the Color Literacy Forums, which are organized by the Color Literacy Project in partnership with the Inter-Society Color Council, that's the ISCC, the International Color Association, the AIC, and Cumulus, a leading global association of art and design research and education. The focus of today's forum is the evolution of color vision. This fascinating topic concludes our 4-part series on color perception that has taken place over the course of the past year. Before we begin our program, though, I want to invite everyone to use the chat box to tell us who you are, where you're from, so your location or university or professional organization, so we can learn about each other in this international collaboration. So thank you. So everybody, please let us know who you are, and it also gets your fingers exercised for putting questions in the chat box, because we want you to know up front that this is a conversation between everyone here, and we want to be able to respond to everyone. The Color Literacy Forums are a virtual platform that feature presentations and interactive conversations at the post-secondary level, which includes university, industry professionals, and the public at large. The long-term goal of the Color Literacy Project is to build a 21st century paradigm for learning about the role of color in our lives, based on an interdisciplinary STEAM model that integrates sciences, technology, engineering, the arts, and math. This new model aligns color education with current needs in the culture, provides cutting-edge resources, and offers dynamic networking opportunities for all stakeholders. So fluency with the language of color sharpens our sensory perceptions, enhances our ability to communicate, and connects us to the world. And we will really be seeing that today. 21st century color literacy isn't just for scientists, artists, and designers. We believe that it is for everybody. Before I introduce our 3 guest speakers, and we're very excited to have our speakers today, here's a brief overview of our program. It's a 90-minute program. We're doing the introduction now, then each speaker will give a 15-minute presentation. During that time, please write any questions you have in the chat box, because after the 3 presentations, we will have a 30-minute panel discussion. They'll be moderated by Robert Hirschler, and Ingrid Calvo-Ivanovich will be a co-moderator, keeping an eye on your questions from the chat box. So she will be feeding them to Robert as well, and to the panel during the discussion. So both Robert and Ingrid are fellow members of the Color Literacy Project. All right, so let's get on with our program. The title of the first talk, and Andrew, I think you're bringing up your video first. This is introducing Andrew Parker. His talk is The Color Vision Evolution in the Blink of an Eye. After working as an artist and as a designer and after working as an artist and biologist in the UK and Australia, Andrew came face to face with the brightest colors found on coral reefs and in rainforests. He completed a PhD at Macquarie University in Australia, and then worked in the USA at various locations, including the Los Angeles County Museum of Art, Massachusetts Institute of Technology, and the Smithsonian Institution. In 1999, he moved to Oxford University to study the science of the natural technologies behind these vivid colors. He even placed them into the design of products, a subject known as bio-inspiration, and became selected as one of the top 8 scientists in the UK by the Times and the Royal Institution in London. Andrew has written the popular science books In the Blink of an Eye, which I have right beside me right now, and 7 Deadly Colors, based on his light switch theory, which conceives that the evolution of the eye triggered the Big Bang of evolution. So fasten your seatbelts, everybody, as Andrew takes us back in time to over 500 million years ago. Andrew, we're looking forward to your talk. You have the floor.
Thank you very much, Luann, and thank you for inviting me.
And let me just say one quick thing before you start, Andrew, because if everyone could turn off their cameras and mute their mics, except for the speaker, that would be great. So thank you, Andrew.
Great. Okay, no, thank you very much. And yes, as you just introduced, actually, I'm going to be talking about the light switch theory, which is the evolution of the eye and how it changed life on Earth. There. So I'm just sharing the screen now. I hope you can all see that. Okay. So the story begins really with the evolution of life on Earth. So we have to, for the light switch theory, we need to know how life first evolved. So the Earth formed about 4.6 billion years ago. The first life formed around 3.9 billion years ago, which were simple bacteria-like things, which took hydrogen from hydrogen sulfide. But about 2 billion years ago, life took the oxygen from a much more accessible source, which is water. And here we've got some examples of the limestone columns that they built up as they secreted lime, which can still be found today, such as in Western Australia. But about 1 billion years ago, the first metazoans evolved. These are the cells with a nuclei. They went on to evolve into plants and animals. So from between about 1.1 billion and 540 million years ago, the evolution of the metazoans took place. Now, these are the multi-celled animals. So they're all the animals we have today, which can be split up into about 37 groups called phyla. So between about 1,000 million years ago and 540 million years ago, this evolution took place, and they all evolved, all these different body parts. Now, a phyla is defined by its internal body plans, not the way it looks externally. So it's things like tissue types and the number of layers of tissues, the way the blood and a digestive system is organized. Here are some examples. So the arthropod, you've got insects and crustacea. We're in the chordates, along with fish, amphibians, reptiles, and birds. So what had happened then? We'd got this evolution of these phyla that had been completed by 540 million years ago. But the problem is they all had soft bodies. They were all like a worm of some description or jellyfish, because that's a very convenient form. You don't have to spend much energy on maintaining such a body. But we know life today is filled with hard parts, skeletal parts, shells and spines and bones, which require much more energy and a much more complex lifestyle to maintain. Now, this all came about, these hard parts all evolved at around 520 million years ago. And we don't know why, but we do know it happens because when hard parts evolve, they leave impressions in the fossil record, and we get a much greater fossil record from that point onwards. So this is the evolutionary tree of the body parts. We've got this explosive event where all the different phyla evolve their hard parts at the same time. And this is what the evolutionary tree would look like. You have, I've shown 9 worm-like phyla there, which each independently and simultaneously evolve their hard parts, leaving the fossils that we know from the Cambrian period. There are actually 37, imagine, well, 34 of those in that case. So the big question is why? Why did they do this? We have some explanations put forward, but these explanations refer really to the evolutionary branching that took place much earlier, not the explosive events of the hard parts. And once they got their hard parts, they could swim and they could bury into the sand and do all sorts of things that just as animals do today. Well, I came into this actually studying a group of animals called seed shrimps. These are crustaceans. These are tomato seed and shaped little animals, marine animals, that have a shrimp-like body, totally enclosed within a bivalve shell. You can see in this picture that they have a well-developed they also have chromatophores to change color a little, but they have a transparent shell, which I've been studying recently, actually, in Florida. In Florida also, they produce really nice patterns of bioluminescent light. They produce flashes that each species can recognize the patterns of flashes of their own species. So very nice. So bioluminescence is when you have a luciferin and a luciferase reacts in the presence of oxygen to give off light as a byproduct. So you don't need sunlight here. So this is very common in the dark and in the deep sea, for example. But then I found another thing on these seed shrimps, diffraction gratings, a form of structural color. These are, in this case, surface structures with a periodicity of between about 50 and 500 nanometers. And it splits white light up into its component colors, just like on the surface of a DVD or a hologram on a banknote, for example. And here's the effect. Here's the antenna of one of these animals showing different colors of light from different directions, and a video recording to show that this iridescence or structural color has a function. It functions in courtship. So these are video recordings of a male and female. When the male swims to within view of the female, it releases these iridescent hairs from within its shell, and courtship takes place, mating takes place. Diffraction gratings, though, weren't very well known in nature. In fact, they weren't known at all. But another type of structural color was, which is the thin film and multi-layer reflectors. Here is a single thin film, such as on a fly's wing or a layer of oil on water. The fly's wing is essentially a thin film, and as such, it has two boundaries, an upper and a lower. When light strikes the upper boundary, some of that light is reflected and some passes through the wing material until it reaches the lower boundary. And again, some of the light is reflected and some passes through. The reflected rays superimpose. And when they're in phase, when you have constructive interference, you see a color associated with that particular wavelength. Now, this condition is only satisfied for one wavelength at each position, at each angle. So you might see red at one angle and then blue at another. And that's where we see these common shifting colors on an oil slick or a soap bubble, for example. Well, that was one layer. Typically in nature, we have multi-layer reflectors with at least 10 layers. Now, with 10 layers, you can get 100% reflection. With a single layer, such as the fly's wing, we're only getting around 5% from 5% from each boundary, so about 10% in total. This is a multi-layer reflector from a sapphirina crustacean, a copepod. It's only about half a centimeter long, but it's so bright you can see it even when you're swimming. The multi-layer reflector in this case is embedded within a cell. Usually, though, the layers are secreted by cells and build up as part of a cuticle, for example. They can also occur in fossils where the material is well preserved at a fine scale. This is an 80 million year old ammonite from South Dakota. And here's a 50 million year old beetle from Messel in Germany. You can see there the thorax, head, and jaws. The color is really well preserved. Now, I started looking throughout the fossil record, and the oldest example I found of structural color was in 3 species from the Burgess Shale fossils. Now, these are a very famous group of fossils from the Rocky Mountains in Canada, and they evolved just after the Cambrian explosion event. So they tell us the end result of the Cambrian explosion. They show a picture of what life was like. This is a worm-like animal called Canadian. It's got these spines along its body. Some of the spines have come free, and if we dissolve away the rock matrix using acid, it'll reveal some original organic material. And you can see a diffraction grating starting to form here. So I recreated that using a laser technique in photoresist, and here's the result. And so that's what these spines would have looked like 508 million years ago in this case. So we have these color reconstructions now. But the question is, why are these animals displaying adapted color in the Cambrian? It's very efficient color, so we assume they have a function. Of course, the answer is the same as animals are displaying color today because eyes exist. But how far can you take back this scenario of animals produce reflecting or displaying color, adapted color, and eyes detecting the signal. So that prompted me to look for the earliest eyes. I mean, eyes evolved to detect the highest intensity of light in the sun's output that reaches the Earth's surface, which this is the sun's output under 3 different levels of cloud cover. And you can clearly see that there's a maximum window there of between about 400 and 700 nanometers, 0.4 to 0.7 microns, with a bit either side. And so that's become the focus for most eyes. So with eyes, you turn these electromagnetic rays into this spectrum of colors. But before eyes, that wouldn't have happened. And by eyes, actually, I'm talking about visual organs that form an image and can extract information from that image. So rather than a simple light sensor, which existed prior to the Cambrian period, which detects, for example, what the direction of light or the intensity, which is up and down in the water column, for example. But then also for an eye, you need a sizable brain to process that information. In most cases, at least, this is an octopus brain, and those huge lobes on the side there are the visual lobes, the optic lobes. Now, eyes today are incredibly successful. Only 6 of those 37 phyla of animals possess eyes. In fact, really only 3 of those to a large degree. Yet over 95% of all species today belong to those phyla. And we had a range of eyes in the Cambrian as well. We have them in the Burgess shell fossils I just introduced. Here is a comparison of a living relative of that shrimp-like animal. And you can see that the zones in the eyes correspond really well. So it probably saw in the Cambrian period just as well as shrimps do today. But the oldest eyes of all that I found, and they're very well preserved. That's the good thing about eyes. They have to be fairly large, so they will preserve in fossils. You can't miss them. And it goes back to the trilobites. These are compound eyes, as opposed to the camera-type eyes that we have. And a whole range of trilobites suddenly appeared around 521 million years ago with eyes. And prior to that, there were no eyes. So the first time these eyes were introduced into the behavioral system of animals, it would have introduced the most powerful sense of all, vision, at one moment in time. So it must have created some kind of chaos. Here's one of those earliest trilobites with eyes. You can see the 2 ridges at the front there, the eyes, really quite sizable. And we think that they evolved from these soft-bodied proto-trilobites. They probably evolved an eye. They could see all the animals around them as essentially chunks of protein. And then evolution would have acted on them to evolve hard parts and swimming capacity to become a predator. We also know that the eye can evolve relatively quickly. This is a study by Dan Eric Nielsen and Susan Pelger, quite a famous one, just computer generated to show that an eye can evolve from a patch of light sensitive skin sandwiched between a transparent window and black pigment in at least a maximum of 364,000 generations. And really, there's a big jump near the bottom there, going from lensless to lens, which is just within 38,000 generations. And that basically introduces vision to the world. So if we look at the evolution of senses, really vision is the one that can be introduced overnight. Smell and touch and hearing, for example, would all be introduced very gradually over time. You don't need that leap of no image and image. So what we have is this is a vision of the world 21 million years ago with the best light detectors of the time. And then 1 million years later, we have this. So we've suddenly got vision thrown into a system where all the other animals then are acted upon by tremendous selection pressures to be adapted to those eyes. whereas before there was no real predation. Then suddenly you've got predators identifying instantly an animal and reaching them within seconds. So all animals needed to adapt, and they did that by evolving hard parts. So the introduction of vision does seem to provide an explanation for why animals all evolved their hard parts at this one moment in time. But I think this study is also interesting because it shows, it gets you thinking about what happens when you suddenly introduce vision to a visionless world. Really quite interesting.
Andrew, 2 minutes. 2 minutes
okay.
left. Yeah, thank you.
So, for example, this light switch theory has been applied to politics and business. For example, here's a case in Scientific American where you think about the transparency that's introduced by these new eyes. Then post-Cambrian, animals, the groups of animals went eyes went into slightly different directions. Some, such as the mantis shrimps, evolved many visual color pigments, so they could discriminate color even better. But once you have eyes, you always have eyes. Really, vision is there, and that's it. We find that we think that color vision evolved more or less with vision as well. We've got fossilized rods and cones in 300 million year old fish and 160 million year old birds, for example, but those fossils are quite rare. But we do think that the advantage color vision would have conferred would have backed that up. Now, as a bit of a sideline, as we mentioned at the start, I also studied art. I really wanted to produce this type of structural color that we could find in a lot of birds of paradise and butterflies. If we look at these butterflies, the ones on the right, the two on the right, are quite shiny and metallic-like, a bit glittery. And that's the type of structural color I showed with the flat multi-layers. But those on the left, they spread their color out into all directions and produce a really velvety hue. It's a really luxurious and beautiful color. And by looking at a whole group of animals, I found a common principle that allowed me to manufacture that on existing machines and called it pure structural color. So pure structural color now I've been making in artworks and displaying. Here it is on flexible materials. It's the world's brightest color, perhaps. And here it is in an artwork, a disc of pure structural color displayed against a natural background of pigments. The intensity really stands out. I've been setting up art exhibitions. So there should be one coming up at Kew Gardens, which was a couple of years ago. Okay, taking a bit of time. But when you compare these against a pigmented background, it's really quite remarkable, the difference. Okay, here we are. This is one at, oh, these are some, sorry, I've jumped ahead of myself here. This is a part of the art show at Kew Gardens where I made the structural color inside a kaleidoscope. And we have another one coming up at there's Kew Gardens. And here's one in Los Angeles at the Sage Culture Gallery and also in Palm Springs. And these are some shoes that Nike made. I worked with Nike quite a lot to develop the technology. And fortunately, Prince Charles helped out to set up a company called Lifescape to help promote this and bioinspiration in general. So you can find this on the website lifescape.com. But I'd just like to end really with the fact that color doesn't really exist in the outside world. Color isn't a construct of the mind. And without that first eye evolving, we would have never have had color in the first place. So thank you very much.
Wow. Andrew, thank you so much. That was absolutely illuminating. And one thing that comes to my mind is just how powerful vision is to the world. And again, how much we can learn from the world by studying color. So thank you so much for that. And we're looking forward to questions. I know that I personally have a million questions for you, and I would love to see your exhibits. We're moving on to our next speaker. Talk number 2 is our speaker is Dr. Jane Neitz. The title of his talk is "In Some Ways We See Colors Remarkably the Same, in Others We Are Remarkably Different." Dr. Neitz holds the Bishop Professorship of Ophthalmology at the University of Washington in Seattle in the USA. He works with his wife, Maureen, to answer questions about the biological mechanisms responsible for vision. They are interested in how neural circuits process signals initiated in the cone photoreceptors to provide vision, control axial growth of the eye during development, and modulate activity rhythms and mood. They use a multidisciplinary approach that includes molecular genetics, electrophysiology, light and electron microscopy, and psychophysics. Dr. Neitz asks us to imagine looking at a vibrant sunset, and he says, while it might seem that we are all witnessing the same breathtaking hues, we will soon discover that the truth is much more interesting. So we're looking forward to your presentation. Dr. Neitz, you have the floor.
All right. Good morning, everyone. It's great to be here. Let me see if I can share my screen. Okay, great. All right. Well, it's a great introduction to think about the fact that really color is something that is in our mind. And, I get questions all the time about differences in color vision. Probably the most common one is people say is maybe my green is your red, or people wonder about why different people argue about colors. And so it's a really interesting thing about the fact that, the ways that our color vision is very different from each other and sometimes very much the same. And some of our insight into differences in color vision come from experiments we did in order to cure color blindness in monkeys. It all started because there's a group of monkeys that come from South America, squirrel monkeys, that were discovered to have red-green color blindness pretty much exactly like what humans have. That is, there are some animals that are colorblind as a species, but these animals have color blindness among certain individuals, but other individuals have trichromatic color vision like we have. So normal trichromatic color vision in humans and other primates is based on 3 different kinds of cones. And this is just a cartoon of a little patch of human retina that shows 3 different kinds of cones. And I'm going to use the names, the common names. So there's the 3 ones that can be called red, green, and blue cones. As you can see, most of the cones are red or green, and a smaller number are blue. Now, people that have red-green color blindness, at least the most severe form, and the form that these monkeys had, they don't have 3 kinds of cones. Instead, they just have 2 kinds of cones. But in colorblind humans, usually they have exactly the same number of cone photoreceptors as normal people do. It's just instead of having two types in the red to green range, they just have 1 type. And so this meant that we had the idea that it was possible to cure color blindness by transforming a subset of these green cones into red cones. And so the monkeys that we picked out in order to study were ones that had blue cones and green cones like this. And then we developed a gene therapy method where we could transform a subset of the cones, the green ones, into red ones. And this was performed by using gene therapy where we added the missing gene to the retina by making injections underneath like the retina, so the solution had access to the cones, and infected a random number of cones and transformed a subset, what were green cones, into red cones. And so what was a dichromatic retina with just 2 kinds of cones was transformed into a retina that had 3 kinds of cones. But the question is in an adult animal, if you suddenly change just their retina, but nothing else about their visual system, does this change their color vision? And of course, we had to have a way of measuring color vision in humans. And we adapted a color vision test called the Cambridge Color Test. And maybe many of you are familiar with color vision tests that there's a series of dots, and then written out in the dots or is a letter or a number or a shape or something like that. And then we ask people if they can see the shape. Well, the monkeys can't do that exactly, but we were able to modify the color vision test. So rather than being a shape, there was just a blob of color that was in a particular spot. And over trials, what we can do is we can move that blob around and make it different colors. And we can also change how colorful it is. And so with this test, what we can do is we can train the animals in order to pick out where the color is. And then we can change the colors and see what colors that they can see. Now, these are colorblind monkeys. And so you might ask, how can you train a colorblind monkey to do a color vision test? Well, the answer is that animals that are dichromats that have just 2 kinds of cones, they do have a form of color vision. They can see blues and yellows. And so in order to be able to train the test, we showed them colors that they could see. And so here's our animal, and the whole idea is that the screen is here, and he can and he's going to be able to pick out the position of this blob on the screen over time. And here he is trained. I'll start the video so you can kind of see what it's like. So this is a color. You can see it looks greenish to us, but it's more yellowish to him. And I said that he can pick out the color, but it's most efficient for him to pick out the color just by touching the screen with his tongue. And then when he gets it right, there's a clicking sound that says you're right. And then underneath it, there's a drop of grape juice that appears as a reward. And you can see that he gets to the point where he can demonstrate that he can see these colors really well, and that he has really good color vision for this particular set of colors. Now, the other thing that we have to understand about red-green color blindness is that for people that are red-green colorblind, and the monkeys too, there's a certain perfect red and a certain perfect green that don't exist. And so that red color looks exactly gray. And so here's a monkey looking at it with our trichromatic vision, but here's how this same scene would look like to a dichromat, a red-green colorblind monkey. And you can see that when he's looking at the screen here, he can't see that red blob at all. And he knows that as soon as the screen comes up, so we're asking, show us where the blob is. And he looks there and he doesn't think there's anything there. So this is very frustrating to him. And the way that we do the experiment is we mix in things that he can see and can't see in order to keep him working. But just for this demonstration, here's a monkey that's red-green colorblind, and we're showing him a bunch of things that he can't see. So I'll start this video, and we can see, if you have normal color vision, you can see this nice red blob here, but he doesn't see that at all. And here's kind of his reaction to it. So when he gets it wrong, he hits a buzzer that tells him that it's wrong. He doesn't get a reward. And he just absolutely, and when he gets it right, of course, by chance, you might try that again. But you can see quite clearly that he's frustrated by the fact that he can't see these colors. Well, Then we introduced the gene therapy, and this is an image. This is a video of exactly the same monkey doing the task after he received the gene therapy. And you can see that there's this dramatic change in behavior. Now he just missed with his nose that time, but you can now see this dramatic difference in the way that he sees color. And so just introducing a set of cones in his eye that are sensitive to long wavelengths has changed his vision dramatically, so things he couldn't see before, he could see. Okay, so the way that color vision works is if you only have one kind of cone, there's only one color, everything would be shades of gray. If you have two different kinds of cones, one in the short wavelengths and one in the long wavelengths, then each cone can serve a particular color. And so that means that there's two different colors. And we like to think about it as being blue and yellow. And so people that are red-green colorblind, they see everything that's blue and yellow and shades of gray. And so here's a picture of the large set of Crayola crayons, but I've changed the picture so this now represents what it would look like for a monkey or a human that has a red-green color blindness. And then here's what the same set of colors looks like to a person that has normal trichromatic color vision. And we did a lot of different tests with the monkeys, and we found out that once they had the treatment, they were able to make all the same color discriminations that people with normal trichromacy sees. So you see that there's this huge difference where you go from just seeing basically 4 different colors, blue and yellow, black and white, to this huge array of colors. There's 128 crayons in a normal crayon set. And the other thing is there's other dramatic differences in color vision. Here's another image that's modified to show what it looks like to someone that has red-green color blindness. And here's what it would look like to someone that has normal color vision. Here's the subway map in Tokyo, again, modified to show what it's like to have, if you have red-green color blindness. And here's the same thing if you have normal trichromatic color vision. So the thing is that's so amazing about this is that we didn't change anything in their brain. And I think that people sometimes think that maybe one person's brain might be wired differently than the other, so that the green cones are wired differently than the red cones. So one person would see one person's red would be another person's green. But we can see that actually the messages are going from the cones up to the brain, and it's really a matter of learning what those messages mean, basically by having experience of with the world. And so the idea that our brains would be wired differently, that can't be true. That the differences in color vision really are occurring about what we've learned with the world around us. Well, another huge difference between different humans, not only do some are totally missing one class of cones, it turns out that there's a huge variability across different people in the ratio of red to green cones. And here's examples. These are actually images from human beings that shows the arrangement of different cones in different people. And someone down here, the BS, has lots of red cones but not many green, and this person has the opposite. And you might expect that this huge difference in people's arrangement of cones would make for differences in color vision. In order to be able to see if this is true, we wanted to test a particular capacity for color vision. And that was a capacity where we asked people what color in the spectrum looks purely yellow. So if I have people in the audience look up and down the spectrum and find a place on the spectrum that looks neither red or green is in pure yellow. I see something right here. And then what we did is we took these people that we knew had very different ratios of red to green cones, and we measured their unique yellow. And it turns out there's almost no variability. And there was people that kind of proposed that maybe there would be huge changes in people's color vision when their retinas had different numbers of cones, that turned out to be not true at all. So the question becomes, how is it that people can have such differences in their eyes, and yet have so much the same vision? And we said, maybe it's because, that really our color vision is based on our experiences in the world. And if someone had a different experience, maybe that they would have different color visions. So we had this idea of had people wear colored glasses. And here's an undergraduate working in the lab wearing colored glasses. And we had people also wear colored contact lenses. And then the idea is they would wear these for a period of time and see if just changing your environment of what it is that you're seeing in the world by putting on colored glasses, if that would change your color vision. So we did an experiment where we had people wear those colored glasses or contacts starting at 9 o'clock in the morning until 1 o'clock in the afternoon. And then they took off the glasses, and then they weren't wearing anything colored until the next morning at 8:45. They came into the laboratory. And the reason we did this is because we know that if you're wearing a colored lenses and you take them off, there's a moment where your color vision is different, but it's not long lasting. And we wanted to make sure those results were all washed out by the time that the experiment was over. So
Jay, 2 minutes.
okay, thank
2
you.
minutes.
And here's the result that we, first of all, we had a baseline where we measured people's, this is the wavelength of unique yellow. Then they started wearing the glasses. And after 1 day of the treatment, maybe their color vision changed a little bit, and 2 days, maybe each one of these dots is 1 day. But over a period of time, people's color that they saw as unique yellow changed quite a bit. And then we stopped having them wear the glasses, and still their color vision remained changed for some period of time. Eventually it came back down again. And then we tried green lenses, same thing happened. And so we could see that we could change people's color vision just by changing the world around them. And just to show you how it's subtle. So starting off, the person's unique yellow would be right here. And then after wearing the red glasses, then now clear down here, a color that at least I see is clearly just a little bit on the orangey side, now look like perfect yellow. So we were able to change people's color vision just by their environment that much. And so what we can see is that there's these dramatic differences between colorblind and normal people. But on the other hand, there's differences in people's vision that in their visual systems, but they're very much the same, but it's really manipulated by our environment. And then the very last thing that I talk about is that there's this, that one of the color, there is a color that people have a huge variability in, and that is the color that looks green. People have a lot of, there's that, unlike unique yellow, where everybody, at least in the same environment, looks the same, people have very big differences in the way that they see green. So that's where we get so that in some ways we're very different because of our genetic differences. Some ways, even though we're very genetically different, we're very much the same. And once again, there's still differences that are genetic, even among normal people, that produce huge differences, especially in the way that we see green. And with that, I'll end my talk. Thank you.
Thank you so much, Jay. That was absolutely fascinating. And I feel like will generate so many questions about how that can impact us going forward, and what that really means for how we see color. And I have a feeling that we're going to get a little bit more on that topic as we move into our third and final speaker for the forum. And just before I introduce Dr. Jameson, I want to mention that there's been a few questions about, there is a recording. We are having the session recorded, and we will have that available on the Color Literacy Forum, Color Literacy Project website, as we do for all of our forums. And also another question, there is a 30-minute panel discussion with our speakers. It's moderated, and that is right after our third talk. So with that, I'm thrilled to introduce our third speaker, Dr. Kimberly Jameson. Her talk is titled, Is Human Color Vision Scalable? This talk shines light on the ways that color vision is continuing to evolve and the implications of this research on our lives, drawing parallels to non-human primates and delving into the possibility of tetrachromatic vision in humans. Kimberly A. Jameson is a cognitive scientist conducting research at the Institute of Mathematical Behavioral Sciences at the University of California, Irvine. Color plays a prominent role in her empirical and theoretical work, which includes research on the mathematical modeling of color category evolution among communicating artificial agents, individual variation and universals in human color cognition and perception, empirical work on the genetic basis of human color perception, and the correlated sensory consequences, and comparative investigations of the ways the world's cultures name and conceptualize environmental color. Dr. Jameson directs the Color Cognition Laboratory at UC Irvine and is the lead researcher responsible for the preservation, transcription, digitalization, and public access dissemination of COLCAT research platform that includes Robert E. McLaury Cross-Cultural Color Categorization Archive, which we'll have to hear more about. With that, I'd like to turn the floor over to you, Dr. Jameson. You have the floor.
Thank you, Luann. Let me see if I can share my screen. And can you see my slides?
Yes, but not the full screen yet.
Got it.
Perfect.
So I want to thank the previous speakers as well as the ISCC committee for their participation and their excellent talks and their invitation to participate in this meeting. I want to start with a question following up on the excellent reviews and information we got from Andrew and Jay. And I want to ask the question whether or not human color vision is scalable. And by scalable, what I mean is scalable like a process, model, or coherent system. In other words, I'm asking whether or not the capacity for human color vision is capable of growth and increases in demand, for example. We know already this is possible from what was discussed by Jay and to a certain degree Andrew, because we know that in our non-human primates that color vision has adapted to functions and selections in the environment. And here's an example of how it has adapted in and been scalable in the golden lion tamarin New World monkey that's in Brazil. And we can see that this particular species, like Jay's squirrel monkeys, all males of this species see only blues and greens, but about 40% of the females are also dichromatic and 60% are trichromatic. So over here, over here, the tamarin monkeys, male monkeys see blue and green, but a certain proportion of the females see this particular type of experience, and some of the females see this. So this is a polymorphous species, which means that there's many, or actually there's 6 different phenotypes or visual types in this particular species. And what is this used for? Well, we wonder about why these different phenotypes emerged. Similar to what Andrew discussed, there's selection pressures for that, presumably things that have to do with useful types of functions for the senders and receivers of visual stimuli. One particular function that has been hypothesized is access to food. If you can see bright fruit on a tree, you may get more sugar in your diet. Another one has to do with sexual selection of mates. Maybe you prefer a brightly colored conspecific. And third is detection of predators. So we believe that these particular types of scalability in these primates was in response to let me see, there you are, okay, is in response to environmental pressures. So humans, like non-human primates, also have cone sensitivities that are adaptable, and our typical human phenotype, our typical human observer, has 3 cones classes in their retina. And we know this occurs from inheritance mechanisms that provide this capacity. We know that they're X chromosome linked, and we know that they are also the mechanisms that are related to deficiency, and that's what makes increased scalability possible in humans, okay? As well as decreases in capacity, okay? So you may have heard a lot in media coverage, these fantastical claims about that this is a superwoman thing and this is a woman thing. And really, the main reason why it's trivialized in that sense is because it's X chromosome linked. The basis for these this capacity for color vision is X chromosome linked in primates, and women have primarily 2 X chromosomes. So having more than 1 X chromosome is actually the defining characteristic of being able to have a flexible color vision phenotype. And having multiple X chromosomes or 2 X chromosomes is the defining characteristic of being a potential tetrachromat. What I mean by potential tetrachromat is that you have an observer type that differs from these observer types here that are very well understood. Jay spoke about dichromat males and trichromat females, which have 3 cone type classes in the retina. And these dichromats who are colorblind have 2 cone classes in their retina. And you can also have, by genetic mechanisms, an anomalous middle class or a medium wavelength sensitive pigment that shifts it more towards the long wavelength pigment, and that causes variation in color perception. And for the genetic mechanisms and the perception of these observers are extremely well known. But we don't know very much about the color experience of people who might have this visual phenotype over here in this yellow box. And this visual phenotype is one, as you can see, which has 4 different possible responding cone classes, okay? This is what I'm calling a potential tetrachromat. It's been called a potential tetrachromat in the literature. It's called a potential tetrachromat because there are many gene variants along the genetic array, many substitutions and different types of displacements on the X-linked photopigments. And these additively alter the photopigment peak response to the extent that we actually have all these possibilities and others. And this was known in the early 90s. And when you have these changes or these genetic substitutions, they can be as simple as a single change in one site on the genetic array. In this case, I'm showing you a single nucleotide polymorphism or a substitution for an L-cone amino acid on in codon 3 on position 180. So there's a specific location, one singular substitution where you take alanine and instead you put in serine, and it changes the peak of the responsivity of the cone class. And there are 15 sites where the M and L gene differ. This changing these proteins at each of these positions gives you a range of different possible sensitivities for a pigment class. This was known since the early 90s, okay? So the fact that the M and the L genes are so similar and the way that they're situated on the genetic array allows for these kinds of crossover events and recombinations to be very common. And this is why we have this potential for having these extra pigment classes along with the 2X chromosomes that a female has. I can talk about how that happens on the X chromosomes, but I'll save that perhaps for the discussion section. But people often ask, how frequent are these potential tetrachromat genotypes, given that there are several different ways of doing that in the population? And we know that it depends on the genotype that you're asking about. But for the population where we know that the incidence of male dichromacy is around 8%, we know that the frequency of women who are expected to have nearly equal populations of these hybrid or these potential tetrachromat pigments is about 47 to 50%. Okay, so it's not trivial. What we do also know is that there are different ways to have a potential tetrachromat genotype. One way is to have the normal complement of typical trichromat genes, but also possess an extra variant at this single L cone location that I pointed out earlier. That's a SNP position where you have genes expressing 4 classes: short, medium, long, and what we call an L prime, okay? Or a long, an intermediate pigment. There's another way of having a potential tetrachromat genotype, and that's if you're a mother or a daughter who is a carrier for a deuteranomaly. And these are expressing the typical trichromat pigments plus a hybrid M to L pigment. Now, the reason there's 2 different kinds has affected what's studied in the literature. There's a group of scientists in the UK which have studied these carriers for deuteranomaly. It's headed by Gabriella Jordan, and they studied this particular type of potential tetrachromat because even though it's less common in the European population, it's got a larger spread between the extra pigment classes. And so that would increase your probability, presumably, of having an effect of a perceptual difference. The other type that's studied has been studied by my lab and primarily, and this is one that has a single substitution, which is more common in the population. So it's easier to locate these potential tetrachromat genotypes, but it is a type of a change which only alters the sensitivity of the cone class by 4 to 5 nanometers. So there's trade-offs, and these both have been examined empirically to see whether or not having the extra pigment class matters. So in the studies that I've run, I've looked at subjects with this variant of an L cone as well as this variant, okay, where there's only a 4 to 5 nanometer shift. And in studies that have been done by Gabriella Jordan and colleagues, these are just 2 of their studies. They have several. They've studied hybrid pigment where the shift is larger. Okay, now it's important to say that beyond what we've already shown, that there are several genotype configurations that provide a basis for potential tetrachromacy, and that some of these potential tetrachromats are considered to be obligate expressors of the 4 retinal pigment classes. We have to ask the empirical question, is color experience significantly different for these individuals who have more than 3 photopigment classes in their retina? It could just be that they're just sort of like collapsed into a single type and it doesn't really matter, and there isn't enough of a neural signal to make the difference that's needed to have a perceptual difference in the neural coding. So it's important to note that gene sequences are not enough to produce a human tetrachromat richer color experience. What is also needed at a minimum is a physiological expression of a large enough population of these extra class of photoreceptors, a significantly complex neural wiring system to support the additional signals from these extra cone classes. And as Jay suggested, we think a systematic need and use of an enriched color code, maybe like if you're an artist or a designer, needs to be established through perceptual learning. But all these latter points that I raised are things that are still the subject of empirical investigation. So the question of whether we have an analogous change in perception like we do with these color observer variants is still unexplained fully. We know that we have observers that do have consequences in their perception, but there's a lot of research that we need to do to understand what specifically those consequences are and how they differ from the normal variation we see in the trichromat observers that are in the population. Now, spoiler alert here, because I'm going to give you a couple empirical results and just an example. We do know from research, primarily the studies that I cited above by Jordan and my own lab, that some potential tetrachromats do exhibit color perception that suggests that they have more than 3 functioning photoreceptor classes and that these classes are impacting their color vision. So presumably we should be able to produce a simulation like this for a tetrachromat or for a specific type of tetrachromat, because as I suggested earlier, there will be different effects on perception depending upon what type of potential tetrachromat genotype you examine. But what about the impact in the world? What can we say about possible applications that might be influenced by having a potential tetrachromat genotype and functional tetrachromacy? Well, let's look at 3 examples, practical examples, to understand how these types of phenotypes might affect designing of user-centric environments or applied industrial design. We know
Kimberly,
from
sorry,
our,
two minutes. Thank you. 2 minutes.
Thank you. We know from experience that when you look at environmental scenes under different light bulbs, like an LED light bulb versus an incandescent light bulb, these colors, even to our normal trichromat eyes, can look different, okay? And we know that the CIE is interested in developing light standards, and they want to produce LED approximations of incandescent bulbs because people love warm incandescent illumination, but incandescents are very energy hungry. So a question that the CIE might ask is, do such LED approximations provide good color fidelity for all observer phenotypes, including these potential tetrachromats? And so we can run an experiment where we do a minimized flicker task, presenting illuminance and scenes illuminated under these 2 different spectral emission spectra from 2 different illuminants, an LED approximate of an incandescent and an incandescent light, and we can ask whether or not subjects exhibit color matching. And we can look to see whether or not this phenotype here gives a different set of color matches or less color fidelity under the LED approximate than you see with this normal trichromat. And you might expect that because the difference that's occurring, the large difference in the spectrum, will be impacted down at this medium to long wavelength end of the spectrum. So that's one possible application that we can look at. Another one that has more to do with translational types of concerns is, what is it like? How could we communicate what a tetrachromat observer sees compared to a trichromat experience? So to capture and communicate a tetrachromat difference, we might look at simulating naturalistic images with a potential tetrachromacy filter. So here we look at a picture of a sunset that we all can appreciate, and we can analyze it pixel by pixel to study with a heat map the differences that are seen between the trichromat processing of each pixel of this image and a tetrachromat processing. And what we can see is that a tetrachromat will see more energy or stronger percept in some parts of the scene and a weaker percept in other parts of the scene. So we can look at how these particular types of simulations tell us the difference, the sort of the subjective qualitative difference in the qualia between these two observer types. Otherwise, it's difficult to know. Okay. We can run a painting experiment where we have observers take a fixed palette and sit down and match Munsell color chips from the Munsell color checker, and we can measure these stimuli, and we can look at the data set of measured reproductions that are produced by these artists, and we can ask whether or not a trichromat artist needs more basis functions to describe their data set compared to a tetrachromat artist. And it turns out when you apply machine learning to that type of data, then we find that these types of emergent eigensolutions or basis functions fall out of the data of the measurements of these color matches for these tetrachromats compared to a trichromat. So there's lots of questions we can ask about applied size of this particular phenotype. And in particular, as Jay mentioned, there are the clinical applications which involve tapping into the neural flexibility of adult sensing observers and getting a dramatic change in their percept in a short amount of time when given the opportunity to use those cones, those extra cone classes. So I think that in summary, we know that human X-linked color vision genetics continues to be scalable despite the limiting conditions that are in place due to ocular physiology, our neural coding constraints, and the visual processing trade-offs like acuity for color signal. We think this is the case, that it continues to be scalable in humans, that human potential tetrachromat color perception is associated with X-linked photopigment phenotype variations, and this has been shown perceptually, but more work is needed to understand exactly what it means. And that existing models of human color processing can benefit from incorporating this information. And this is already seen in recent publication and utilities provided by Andrew Stockman and colleagues for generating cone spectral sensitivities, even with mutations occurring of the kind that I described. And we should keep in mind that developing applied technologies like these translational clinical tools and design approaches, we may want to consider in our applications the relevance of this potential tetrachromacy phenotype. And with that, I'll just thank my main colleagues in medical genetics at UC Irvine, Dr. Kinney and Sherry Adelano, who work with me on this particular genotyping project. Thank you.
Thank you so much, Kimberly. And thank you to all our speakers, to Andrew, to Jay, and to Kimberly. And this opens up our next part. So Kimberly, if you would not screen share now, and we'll have going to have a panel discussion. It'll close at 12:30 Eastern. So let's see, we're going to bring up the cameras for Kimberly, for Andrew, and for Jay. Also, the moderators for this component of our event today is Robert Hirschler of the Color Literacy Project. I'm going to turn over to Robert momentarily, and also to Ingrid Calvo-Ivanovic, who has been monitoring your chat box. So between you all, I will turn it over to you, and thank you so much. So Robert.
Thank you very much. First of all, thank you, all three of you, for these fascinating talks. I mean, it's really breathtaking, and it was an arch from the very beginning of I appearing in the world to the maybe maybe call it the future of tetrachromacy in humans, and all the problems in between, of which Jay spoke. Obviously, the three of you know each other well and know each other's work, but I would very much like to hear what you want to hear from each other after each of the presentations. Any questions from any of you to any other of you before we open the floor for the audience questions?
Sure.
Jay?
Sure. I have a question for Andrew. So I read your book and just your talk now. To me, this whole idea that the Cambrian explosion is caused by the evolution of the eye just seems like it has to be true. It just doesn't seem like it could be controversial. Are there alternatives that people want to offer, or is this pretty well accepted?
I think it's becoming well accepted, really. At the start, it was introduced in a field that didn't really know a lot about vision and didn't really consider vision anything special to the rest of the senses, I think. And that's the thing when you consider the importance of vision today and the power and how everything has to be adapted, because unlike other senses, if you don't make a sound, you can't be heard. But with vision, wherever you are, you're going to leave an image on a retina. So you have to be adapted, and your whole body has to be adapted, your behavior, your color, your appearance. If you're a stick insect, you have to sway in the wind like other sticks. And the whole body is just totally adapted to vision. And you can go on and on like that. There are a few exceptions, with senses like echolocation in bats, but they're very odd, they're very unusual things, and they occur in a world where everything is adapted to vision as a basic thing. So you suddenly throw that in one moment in time, and it's going to create chaos. And I think that's the most important thing, really. And if you have the change between the evolution of vision and not, the step before, really, it's not 50% or 60% of the way, it's at 1%, and then you suddenly make this leap with a lens to 100%. So within almost overnight in the geological scale, you make this leap, and it's just going to have this tremendous effect on animal life in general. Yeah, so I think now, I think the other theories put forward really are more explain the cladogenesis that took place, the evolutionary branching of the different phyla. So changes in oxygen and carbon dioxide levels, for example, which fluctuated. They went up and down a lot during before and after the Cambrian period. So yeah, I like to think of it more as how powerful the introduction of vision would have been.
Well, I'm a fan of the idea.
Oh, no, that's great. Thank you very much. And
I have a question.
Yes, Kimberly, please.
I have a question for Jay. I know that he's been working on a Cambridge color test, and I wonder how that's going for tetrachromacy or potential tetrachromacy.
That's I have worked on that a little bit, and it's an interest of mine, but it's mostly been on the back burner. I love the idea that I think that if we lived in a world that, even with our trichromacy, if we lived in a world that everything was just black and white, there was no color, and then all of a sudden, things became colored, would we see those colors? And that's a question that I have, but I kind of believe that it takes experience with the colors. And our world is very much, most of the color that we see on screens and paints and everything are based on mixing of three different primaries. And so we kind of live in a world that isn't really suited for somebody that has an extra photopigment. And so I think that maybe part of the reason that some of the people that are known to have an extra photopigment don't accept extra color vision is that they just, it's like a person that had full color vision but never saw color because they lived in a black and white world. So I like the idea of having a really good color vision test that would be like the color vision, the Cambridge color test, but would have another color, and then be able to try to show people stimuli that would stimulate their extra dimension of color vision and see if we couldn't make it stronger than it is. So that's my kind of fantasy thing, but I haven't gotten very far on it.
I think a good starting point for that is Jordan and Boston's study of CD829, which they did find pigments where you had a separation. So it's difficult to do, which is why I wondered if you were able to make any traction on that. But Jordan and colleagues did that with CD829, which was really a remarkable finding, in my opinion.
Yeah, I don't have any problem finding people that have good, I mean, we can do genetic testing and find people that have good separations between their cones for this kind of an experiment, but the rest of it takes some work and we really haven't made progress on that.
Ingrid, do we have an interesting question from the audience?
Yes, we have a question for Kimberly. It says, it's from Jude Mooney. It says, do we know what percentage of trichromatic women see more red?
That level of investigation has not been done yet, for two reasons. The first reason is, as Jay was suggesting, we can find lots of women who have the genetic potential to have an extra pigment class, but we don't find many who seem to be able to enlist that in perceptual functioning. So they're not demonstrated as what we call strong tetrachromats or functioning tetrachromats. They do exist, which is an important result because it tells us that that is possible, and it gives us room to move in a modeling situation for developing observer models. But in terms of doing a survey of sufficient numbers of individuals who have this functional tetrachromacy, that particular result isn't in hand yet. So like I said previously, it's important that we keep in mind that you need the neural programming, you need the perceptual learning, you need the genotype, you need a whole bunch of stuff to be satisfied in order to get functional tetrachromacy. And that is making it difficult to get enough subjects to make a concrete study of what the phenomenological differences are in color, in everyday color perception. So no, I don't know how many potential tetrachromats see more red.
But may I ask you, have you heard about, there was on the internet quite a bit of news about a lady who is a strong tetrachromat and paints and has all kinds of strange colors in her paintings. What do you think of it?
Well, that participant has been my subject for over 10 years. And actually, I think she originally contacted Jay for genotyping and then followed up with me because I was more available to carry out our research. But she is a confirmed, has the right genotype, one of them. And you know, we've been teasing out what the differences are. And, you know, some of the differences that I've seen have been small local differences. So it's important to keep in mind that this may not be like a huge dimensionality change. It could just be something as simple as being able to see more intermediate steps in the Munsell Book of Color and accurately and reliably repeating them. Okay, not just seeing it like, yes, I see a step between, you know, Munsell value 5, you know, chroma brightness, this and the other one next door, but I actually can reproduce it. So in my studies I've done with painting, I ran her painting reproductions alongside other artists who were trichromats. And that was the tetrachromat basis functions that I showed you in my talk that emerged from her measurements. So that's an objective measure. And the hard part is getting beyond the subjective reports of the participant when they are so compellingly sure of themselves that they have these capabilities. You have to sort of find a way to get at it without asking them and do it objectively. So that's what we've been trying to do. And you know, she's one observer. You know, are there others? Well, I think CDA 29, when Boston and Jordan used pigments, which they designed from this observer to be distinguishable by her, but not her carrier sons, her normal sons, then those dimensions popped out as reliable discriminations. I think those kinds of results are important. But once again, they have to be individually designed. They're not ones that we're going to be able to globally find what is the difference for all potential tetrachromats, because it's going to be an individualistic type of thing. So it's scalable, but it's not robust and conserved like trichromacy is. Okay, it's, in my opinion, it's sitting there waiting in case it's needed. And you know, maybe in the case of artists, they cultivate enough perceptual learning that they find a use for it. I don't know. It's still an open question.
Any other question from the audience, Ingrid?
Yeah, we have a question for Jay. It's from Robin. How does opponent processing change when the dichromat monkey becomes a trichromat?
That's a great question. And so just for the rest of the audience about the question is that it's well accepted that at the level of our cone photoreceptors, there's 3 different kinds of cones, but there's a next stage where there's parts of our nervous system that are comparing the outputs of the different cones. And in people that have normal trichromatic color vision, there's 2 different comparisons. There's one that gives us blue-yellow color vision, and a separate set of connections between the cones that give us red-green color vision. And the reason, and one thing that makes us know that it's opponent is that if you mix blue and yellow together, you don't get a color. And red and green, there's no reddish green; those cancel each other. So that's why they're called opponent. And so at one stage, there's the cones, and then there's an actual nervous system component where you get this red, green, and blue, yellow circuitry. And to start off with, the monkeys only have circuitry that's basically the basis for blue, yellow color vision. And then, at least from their behavior, it looks like that they must have a second opponent dimension, because their color vision is very, very much like what we see in, at least their discriminations are very much like normal people. So somehow they are able to make an opponency between the red and green cones once they have them. So that's a huge, but how they do that, I don't know. That's a huge question. I really like to know. It's something we're working on, but I don't really know.
There was an interesting question to Andrew from Robin about the earliest colors seem to be due to structural color. Can you further comment on the relative importance of structural color or iridescence versus pigment color in driving the evolution of color vision?
I think that probably there would have been pigment color as well in the earliest post-eye animals. It's just that pigment doesn't show up well in the fossil record. It tends to break down, whereas quite often with fossils you have replacement materials. other minerals come in to replace the original organic parts. And so pigments in that case are lost, but structural colors being structures, if the preservation is fine enough, those structures will be replaced by other minerals, but retain the same dimensions. So you can calculate what color they would have reflected. So we would know more about that. But in general, structural color is used more for advertising oneself, advertising armory, sexual signals, as opposed to camouflage, really. There's an interesting case, actually, that's different there is beetles. A lot of beetles, it serves them well to be green, to camouflage against a green leaf. But green pigments are quite rare in animals. So to achieve green, beetles, which is one of the commonest group of animals in the world, there are thousands of these cases. They all evolve a yellow pigment in combination with a blue structural color to produce green. So all the green beetles, just like green parrots we see, are not green at all, but purely blue, and a blue structural color and yellow. All the green amphibians too, and reptiles in that way. So the difference between trichromacy and tetrachromacy and dichromacy would become interesting there.
Any other question among yours in the panel, or comment to each other?
I wonder if I can just ask Kimberly and Jay, when primates evolved trichromacy, what do you think the purpose of that was? What was the driving factor? We're often quoted that they can discriminate between ripe and unripe fruits, but then that has been questioned more recently. I wonder if you have any thoughts.
There's a lot of argument about that. And I did my PhD with one of the people that, it's the first, Jerry Jacobs at the University of California in Santa Barbara. And he is one of the early people that studied primate color vision. He and his PhD advisor. And so he because people argue, they go, oh, it must have been fruits, or it must have been this, or it must be that. I mean, one of the other arguments is that having trichromatic color vision is good for reading the color of faces. So there's social cues. So if somebody's embarrassed or angry, their face turns red, so forth. And Jerry says he thinks that color is good for all of these different things, and they probably all work together as advantages that drove evolution. I kind of like the idea that, we can think of so many different ways that, being able to see color, would be useful. And I think that they might all provide some amount of selective advantage. So I don't know that any of the things were sole drivers of it.
Kimberly, any thoughts on that?
Well, I would agree. That was the first slide I put up of the golden tamarind, which suggested there's multiple factors involved, possibly. Sexual selection is the social signaling that Jay was commenting on. So Changizi has done some work early on that primates with faces that have exposed skin as opposed to fur and exposed private parts can socially signal blood flow and arousal by tints on the skin. Okay, just like when we blush, it signals something with our emotional state, and people have argued that that may signal receptivity for conspecifics, and they get the advantage in terms of the choice of conspecifics to breed with. There's lots of simulations out there on detecting predators in camouflage, as well as detecting carbohydrate-rich fruit in foliage and young foliage. So probably a multiple of factors contributing, all pushing and converging to select for the development of trichromacy. And it's important, from what Jay suggested. Jerry Jacobs showed that trichromacy emerged a couple different ways in New World and Old World primates. So it happened twice independently and in not the exact same fashion. So that tells us it was valuable for something, evolutionarily. And I think from my perspective, the way to think about it is if a capacity is sitting there, evolutionarily, it'll get used. Like Jay's squirrel monkey, Dalton, used the pigment that they gave him. And if there's pressure for it, it's going to get used. But the dynamic and properties of the visual system allow it to continue to evolve, I think, is the other important point.
Thank you very much. I'm sure that we could go on, and I would love to listen to you for hours because it's fascinating talk. But I'm afraid that our time is up. So it's back to you, Luann.
Oh, thank you. And thank you so much to everybody. For all the audience, join me in a round of applause. Virtually, thank you so much. This was brilliant. And as I said, for vision is so powerful in the world. It is really what it compels the Color Literacy Project forward. So Ingrid left a link in the chat box. That Ingrid, did you want to say that just about the recording?
Yeah, we have in the Color Literacy Project, we have a YouTube channel, so you can rewatch this edition of the forum and the previous ones. And I left the link there on the chat box. Also in our website, we are always publishing the forums we organize.
Yeah, so we really encourage you to check out the website, and this is an evolving project. We're so thrilled to have so many people join us today, and we are aiming for the next forum to be at the end of January, so stay tuned. And once again, thank you so much to everyone, and have a very colorful rest of your day. Take care.